from Dr. Chen, and he's going to talk about the development of gene replacement therapy for CMT4A demyelinating neuropathy. Thank you.
Thank you, Catherine, for a nice introduction. And then thanks. Oh, sorry. Thanks for the Research Foundation to invite us to present our work on CMT type 4A. So CMT4A is an autosomal recessive, severe phenotype, early onset. It is the peripheral neuropathy, obviously, with reduced nerve conduction velocity. It's mainly demyelinating neuropathy, but I think it still has some axonal component involved, especially at the late stage. And unfortunately, there's no approved treatment for this disease, and which is caused by loss of function mutation in GDAP1 gene. GDAP1 stands for ganglioside-induced differentiation associated protein 1. The gene of GDAP1 is located in chromosome 8, transcribed into a mRNA with 6 exons, and translate to a protein with 358 amino acids. The expression of this protein is ubiquitous. The location of the protein is in mitochondrial membrane. Function of the protein involves the mitochondrial dynamics, including the mitochondrial fission, mitochondrial respiration, transport, and mitochondrial energy production, as well as oxidative stress response. We hypothesize that the lumbar intrathecal delivery of normal copy of GDAP1 with AAV9 might ameliorate disease phenotype in GDAP1 mouse model. To test this aim, we designed the vector, AAV9 mediated GDAP1 vector, and tested safety in this vector in white type animal, and also in parallel test the efficacy of the vector in GDAP1 mutant mice. So this vector basically includes a weak promoter here, driven codon optimized the human version of GDAP1 gene, flanked with 2 ITRs, and packaged the AAV9. And then we checked the quality of the AAV9 on alkaline gel, TEM, as well as ELISA. And then we also did an in vitro cell culture study to see this vector can express well in HEC293 and LEC2 cells. And then we went on to move to the safety study where we dosed the animal, white type, black 6 animal, intrathecally with vehicle, low or high dose at postnatal day 42 to 60 days or 56 days. Following the treatment, we monitor the animal longitudinally for safety by observing any neurological abnormality, survival, weight in the body weight. And at 3 weeks post-injection, we utilized the 4 mice each group for a GDAP1 expression and toxicity panel, serial toxicity panel. At the end of experiment, when mice reaches 12 months post-injection, we utilize all remaining mice and for a detailed organ toxicity evaluation by a vet pathologist. And I don't have time to describe all this data, but I just want to point out this study seems safe. We saw a GDAP1 expression inside nerve with no side effects, no any effects in body weight or survival. All animals survived till the end of the study. Administration of the vector by intrathecal delivery seems safe and well tolerated. Overexpression this protein with weak promoter seems not a concern, no safety concern. So in parallel to this safety study, we performed an in vivo efficacy study in GDAP1 mutant mice. We dosed the animal also intrathecally with low, mid, and then high dose. And we dosed two different animal age. One is postnatal day 7 to 10, and the other one is P120. We monitor the mouse safety parameters. We also did a longitudinal battery of behavior tests, as well as nerve conduction velocity. And then at 3 weeks post-injection, we used 6 mice per group and did an RNA-scope to verify the GDAP1 RNA expression, serine toxicity, as well as serine biomarkers, and also performed the histology inside the nerve. At the end of the experiment, where mice reaches 24 months old, we euthanized all remaining mice for a further detailed toxicity evaluation. So now we can, that's back. Okay, so now we actually get to the results part. So this figure showing on the left, showing with dose-dependent GDAP1 mRNA expression following treatment at 3 weeks post-injection. And then the panel on the right also is just showing the quantitative data of our treatment. We didn't see any effects on serine toxicity panel, including AST/CK-BUN, indicating our treatment didn't generate any toxicity in liver, heart, as well as kidney. We checked the bone as a biomarker, potential biomarker indicated in type 4A, like neurofilament light chain and cellular adhesion molecule 1, but we didn't see much of phenotype as well as treatment effects. So we went on to measure the nerve conduction velocity at different ages, starting from 13 months old. We saw a reduced nerve conduction velocity in these mutant mice, which is improved by our treatment on the right. And then we did a look at the behavior test results, which here shows the reduced locomotor activity in our mutant, which again showing improvement with treatment. Similarly, we performed a load rod study, and then this time we see this load rod deficits only in female, showing in red on left, but not in male, showing blue in the left. And then this deficits in female also can be improved by our treatment, especially early and middle dose. And then to summarize the efficacy study in this mouse model, we saw human GDAP1 mRNA expression inside the nerve. We didn't see any serine toxicity panel, and no effects on serine biomarker, including NFL and CAN1. And we saw improved nerve conduction velocity, but we didn't see much of change in map amplitude. We saw improved behavior phenotype in locomotor and as well as locomotor load rod performance. Currently, we are doing a histology using sciatic nerve and spinal cord as well as DRG to further confirm our efficacy results. So current ongoing study and upcoming plan include that we'll finish up the study, obviously, and writing up a manuscript and then putting together a package for our FDA pre-IND meeting. We're also looking for financial support of commercial partner to move this project to clinical. And in the meantime, we also have CMT4A rat model. We are characterizing them now and then setting up a study plan to trying to confirm the results from our mouse study. The other thing we have been doing is exploring better AAV capsid for transducing Schwann cell. So other previous speakers have been touched this challenge as well as opportunity in this field. But in our case, we feel the most important is still the AAV serotype. AAV9 to us seems used widely, still not ideal. So it's not efficient enough to us, at least in certain experiment. We have been trying to develop a better capsid for transducing Schwann cells. Here on the right, we have some capsid variants which show some promising results just by looking at the by distribution in Schwann cell or Schwann cell after intrathecal delivery. We are trying to move this forward by doing more experiment in vitro as well as in vivo small and large animals. The second is animal model is definitely a problem here. For most of our study in CMT, this model is far from optimal or ideal. For example, we work on GAN disease, GAN, just like Dr. Bonham mentioned. We worked on CMT4A, and we worked on CMT4K, which is SERF1 syndrome. All this, we worked first on mouse model, but eventually we also needed to generate or import rat model trying to do that study again. The DRG toxicity is still a concern, but fortunately we use promoter or we use regulatory design or even tissue-specific promoter. We can mitigate this DRG toxicity. IT route administration seems widely used, also can have certain advantages such as avoiding neutral antibody. You can low dose potentially, also circumvent the blood-nerve barrier possibly. So that's pretty, seems to make sense to use here in a CMT disease, but it takes a lot. It's not easy to mass technique because most of the case we dose the small animal blindly. The space there is very small. To avoid the niching the spinal cord, causing any damage in the animal, I need more practice. And in our lab, we have been working on dozens of different disease, neurological disorders, not just CMT, but also other diseases. We have a lot of lessons learned. So funding seems always the issue. Some funding, even we have plenty of funding, we can move this project relatively faster. So with SPG50, we just used 3 years to move that to clinical trial. But for other diseases like AGU, I'm working on one. Even 80 years, we generated preclinical data, but still at the stage of enabling that enabling this clinical trial. So funding is very, very important. The other thing, the treatment, gene therapy is always the earlier the better. But for CMT, I think it's never too late to start because this is a slow progressive disease. You can have a bigger, longer window to deal with it. But treating early potentially give results. As Dr. Bannerman said, their next design is trying to dose younger age patients. Risk, sometimes we need to take risk to speed up to the study, trying to development the therapy. The other thing, obviously, other speakers mentioned, clinical trial readiness, non-human, natural history study, the patient registry and biomarker, as well as clinical outcome development are very key points we need to do in parallel with the preclinical study. So look at this CMT4 type. We altogether, we probably only account for like less than 5% of all CMT cases. But we certainly have 13 different subtypes. Most of that is very rare, ultra rare, very rare. So As the previous speaker mentioned, like Dr. Baldwin, then she mentioned that she working on 4B1, and then Dr. Cleopa worked on type 4C, and then collaborate with Dr. Kat Lads, they published data on CMT4J. We are now working on the type 4A as well as type 4K, which is SERP1 syndrome. We are trying to move A, C, and J, K, ACJ to clinical trial. Our institution have the ability trying to move the project forward. Obviously, we need funding support, but we have clinical team and regulatory team to help us with this process. We have moving SP50 and CON7 to clinical trial in previous years. So with other type of the CCM4 subtype, we certainly feel that with this disease can be taken care of, maybe even better with better knowledge, more knowledge, better and capsid variants as well as delivery methods. I think most of them can be taken care of in the near future. And with this, I'd like to thank my mentor, Dr. Stephen Gray, and our colleague in the lab who has actively involved in this project. And I would like to thank Dr. Burgess for sharing the animal mouse model, Dr. John Sverin, and Dr. Kathleen Fossey from CMT Association for the support and the advice, and Taysha Gene Therapy as well as CMT4A for their funding support. So I will stop here and take any questions if you have. Thank you.